Okay, so for our final chapter that's going to be covered on exam 2, we're going to talk about the immune system. The immune system is a little bit different than our other systems because in our other systems we typically have one primary organ or structure or a collection of them that kind of serves that main purpose. For the immune system, we're mostly talking about a whole variety of different cell types and different molecular structures, and they're kind of scattered throughout the body in different locations, but they work together with the main goal of defending the body against pathogens. Pathogens being any kind of bacteria or virus, anything that can cause harm to us. So infectious agents are organisms that cause damage and can even cause death in the host organism. So in this case, the host organism is going to be humans that they invade. If they cause harm to the host, they're said to be pathogenic. Pathogenic meaning, that they bring harm or disease. There's 5 major categories of infectious agents. You have bacteria, viruses, fungi, protozoans, and multicellular parasites. So we're going to go through a little detail on each of those. Bacteria are microscopic, single-cell prokaryotic organisms. So if you've taken micro, maybe you recognize that term prokaryotic. They can be shaped as one of the 3 following. Cocci are these spherical shapes that you see up top. Bacilli are these rod-like structures in the middle, or spirilla are kind of coiled. So they come in a variety of different shapes. Certain bacteria cause disease, but the vast majority of them do not. So the majority of bacteria are non-harmful. We're just kind of blatantly aware of the ones that are harmful because they make us feel bad. Virulence is the ability of the bacteria to cause disease. You have different levels of virulence. Some of them are more potent than others. The virulence can be increased by the presence of this external sticky polysaccharide. So polysaccharide is kind of like this sugar molecule that forms a capsule around the bacteria, or the ability to release toxins. So if it can release toxins, it can be more virulent or more harmful to you. The next one is viruses. Viruses are not cells. So when we talked about bacteria, we're talking about living single-celled organisms. The virus is not a cell, it's not alive. They're much smaller than bacteria. Structurally, it's a nucleic acid, so either DNA or RNA, enclosed in this protein shell called a capsid. So it's literally just a nucleic acid, either DNA or an RNA, inside a little protein shell. So viruses are what we call obligate intracellular parasites, which means that they have to enter a host cell in order to reproduce. If the virus lands on a table, for example, which is not a viable host for a virus, it's not just going to replicate. It has to gain access into the cell of a living organism in order to reproduce. So what happens is the virus will infect a host cell, and it essentially gets into the cell and takes advantage of all the machinery that we have in our cells, and it uses our cellular machinery to start to make copies of its viral RNA or DNA and its capsid. So it makes all these copies of itself, and then it ultimately causes the cell to rupture, and it releases all of those new viral particles from the host cell, and then they move on to infect other cells. So in order to get rid of the virus, the immune system has to kill the host cell. So our immune system would have to get rid of any of our cells that the virus has infected in order to clear the virus from our body. So examples of viral diseases include the common cold, chickenpox, and HIV. Fungi are eukaryotic cells that have a cell wall outside of their plasma membrane. So the bacteria was single-cell prokaryotic organisms. Viruses are not even a cell. Fungi are eukaryotic cells, and they have a cell wall outside of that cellular plasma membrane. So your fungi includes mold, yeast, multicellular fungi that releases spores. And then you have proteolytic enzymes. Those are like degradative enzymes, and they get released and they cause inflammation, and redness, and swelling, and you can see that in these images. Fungal diseases are referred to as mycoses, and in healthy individuals here in the US, that's typically limited to superficial infections. So your skin, your scalp, your nails, things like that. So ringworm and athlete's foot are good examples of fungal infections or mycoses. Other fungal infections involve infections of the mucosal lining. So vaginal yeast infection would be an example of something affecting the mucosal layer. And then you also have some internal fungal infections. So histoplasmosis, which is one that affects the respiratory system, that's an example of one that's not so superficial. It's definitely more internal. Protozoans are eukaryotic organisms that do not have that cell wall around their plasma membrane. Some infections that can be brought about by protozoans are malaria and trichomoniasis. And then multicellular parasites are those parasites that are non-microscopic organisms. So basically they're large enough to be seen without the use of a microscope. They reside within the host organism. And from there, they take the nourishment of the host organism and use that nourishment so that they may grow themselves. So parasitic worms, such as a tapeworm, gets into the GI tract of humans and other animals, and it uses the nourishment that those animals consume to grow itself. Prions are different than kind of bacteria and viruses because what these guys are just little small fragments of infectious proteins, and they can cause disease in the nervous system. So if you've heard of mad cow disease, this is caused by prions. It's actually called Creutzfeldt-Jakob disease, or sometimes bovine spongiform encephalopathy, or BSE. And essentially that is a disease that can be spread from cows to human if it comes from an infected cow. And again, they're just little small infectious protein particles that can cause pretty bad disease. So they actually quit using nervous system tissue from cows in different feeds to kind of prevent the spread of that disease. So let's look at the cells of the immune system. We've seen some of these back when we talked about the blood. These are leukocytes, or white blood cells. So the leukocytes are the ones that really work to protect our body against these different pathogens. They're formed in the bone marrow, and then they start to circulate throughout the bloodstream and can take residence in different tissues. So we've got our granulocytes. Our granulocytes are neutrophils, our basophils, and our eosinophils. Then we have monocytes. Monocytes are the ones that go off and become macrophages, which are large phagocytic cells. And then we have lymphocytes. So for lymphocytes, we have T lymphocytes, B lymphocytes, or NK cells, which stands for natural killer cells. Now this picture is just here so that you can recognize that we have immune cells that reside in all throughout the body. So we've got specific cell types for different regions of the body. So microglial cells are kind of our resident immune cell in the brain, and then a variety of different types in our lymphatic system. Alveolar macrophage is a specific macrophage that you find in the lungs. Dendritic cells are specific cells that you find in the skin and the mucosal membrane. So just to help you recognize that these guys are kind of scattered throughout the body, and they can take up residence in certain tissues and become sort of specific for that particular tissue. Cytokines are something that's also found in the immune cell to help boost immune responses. Now, cytokines are not cells. They're these small soluble proteins. They're produced by cells of both the innate and adaptive immune systems that we'll get to in a little bit. And they just kind of help to enhance, regulate, facilitate the different immune responses. So similar to a hormone, cytokines are released from one cell, and then they bind to the receptor on a different cell, and then that causes that cell to carry out a certain function. So if the same cell that released it also binds to it, it's called autocrine. So let me clarify that. So if the cell releases the cytokine and the cytokine kind of loops around and binds to a receptor on that same cell, it's called autocrine. So auto meaning self. That particular cell released it, and it also was the target cell. If the target cell is a neighboring cell or a cell right next to it, it's called paracrine stimulation. So one cell released the cytokine, and then the cytokine traveled over and stimulated the receptor on a cell next to it is paracrine. And then if the target cell is a distant cell, so the cell that releases the cytokine releases it into the bloodstream and then travels a distance to get to its target cells, then that's endocrine. So this refers to the target cell in relationship to the cell that released it. So if it's the same cell, that's autocrine. If it's a neighboring cell, it's paracrine. And then if it's a distant cell, that's endocrine. So cytokines do the following things, and we'll see all of these are just kind of general descriptions of things that they do. These will come up specifically as we continue through this lecture. So they serve as a means of communication between the cells of the immune system. So they help the different immune cells communicate between each other. They control the development and the behavior of different effector cells of the immune system. They regulate inflammatory responses of the innate immunity, and they can also serve as quote-unquote weapons to help destroy cells that we need to kill. So these are just some of them. Interleukin, this one will come up later when we talk about the adaptive immune system or the adaptive immune response. This regulates immune cells. Tumor necrosis factor destroys tumor cells. This is probably the only place we'll see this one. I don't think we talk much more in detail about TNF. Colonase stimulating factor stimulates leukopoiesis in bone marrow. So we kind of saw this come up when we talked about the blood. Briefly, I don't think we went into too much description. Interferon, you've got interferon alpha and beta, which are antiviral agents, and interferon gamma is a pro-inflammatory agent. So we'll talk a little bit about interferons later, again in the adaptive immune response. This slide shows you kind of how our immune system is organized. Here's our immune system. It's broken down into two types of immunity. One is innate immunity, otherwise known as nonspecific immunity, and the other is adaptive, otherwise known as acquired. The two systems work together, but they work sort of differently, all with the same common purpose of just defending us against pathogens and infectious agents. So the innate immunity over here is our immediate response to a wide variety of substances. So it kicks in immediately. It also is non-specific, so it's not going to say, oh, I've seen this particular type of bacteria, here's a response to it. It's just kind of this collection of generalized substances and processes to defend the body, but again, not specific for different bacteria and viruses. It's also something that's there from the time you're born, so it doesn't need any further development. Over here in the adaptive immunity, this is a delayed response, and it's delayed primarily because it is specific. We need time to develop a particular response to a specific pathogen. So in this case, it would say, I remember that particular bacteria, and here's the cell I've created to attack it. So it's specific and it's delayed, and it does require the development of new cells that target these specific pathogens. Okay, so going back to the innate immunity, it's immediate, it's nonspecific, and it's already there. So skin and our mucous membranes, that's part of it, because what does that do? That prevents pathogens from entering our body. And then we've got these non-specific internal defenses. Again, we're going to talk about all of these in details. We've got certain cells, macrophages and natural killer cells, that act as phagocytic cells. They can go in and phagocytize or clear out infectious agents. We've got different chemicals, interferons, that was one of those cytokines, complement system, which will come up in a minute. But these are just different chemical processes that happen within our body, again, non-specifically. And then we've got our physiological responses, inflammation and fever. So again, those are generalized responses to curb, the development of pathogens or reproduction of pathogens in our body. So we've got cells, chemicals, and physiological responses. Those are all part of our non-specific internal defenses as our innate immunity. Moving over to adaptive immunity, we've got delayed response because we're building specific defenses that are specific against a particular pathogen. So when you think of adaptive immunity, think of your lymphocytes. We've got T lymphocytes, which is otherwise known as cell-mediated immunity, and B lymphocytes, which is otherwise known as humoral immunity. The B lymphocytes will go on to make plasma cells, which are important because they make and release antibodies. Okay, so another slide about the difference between innate and adaptive immunity. Innate immunity defends us against a variety of substances. It's called innate because we're born with it. It doesn't require any further development. We don't have to be exposed to something in order to be protected from it. The innate defenses include the barriers of the skin and the mucosal membrane, and again, that helps to prevent things from gaining access to our body. Now, once something does gain access to our body, then we have other non-specific cellular and molecular defenses that will kick into gear. And we saw those physiological responses, the fever and inflammation. Adaptive immunity or acquired immunity involves specific T and B lymphocytes. They're going to respond to different foreign substances, otherwise known as antigens, that we get exposed to over our lifetime. So unlike the innate immunity, the process of adaptive immunity takes several days to become effective, even though it's initiated immediately. So we'll start the process. So let's say you just got infected with something. Initially, your innate immunity kicks in, and it does everything that it can do to prevent that infectious agent from kind of taking over your body. Now, the adaptive immunity kicks in immediately too, but it's a slower process and it takes some time. So while the innate immunity is doing everything it can to slow the infection or get rid of it, the adaptive immunity is sort of building, I like to describe it as building an army, because it's creating cells that are specific to that particular infectious agent so that we can specifically target it and kill it. But because we have to go through the process of doing that, it takes some time to become effective. So this slide shows you innate immunity in a nutshell, basically. So innate immunity includes all the body structures that and processes that, one, prevent the entry of potentially harmful substances, and two, respond non-specifically to a wide range of harmful substances once they do get entry into the body. So we basically say that we have three lines of defense: a first line of defense, a second line of defense, and a third line of defense. The first and second lines of defenses are part of the innate immune system. One is to prevent things from getting in. That's our skin and our mucous membranes. It's our first line of defense. Now, if something does get in, if we inhale, ingest, if we cut our skin and things gain access into our body, now we go into our second line of defense, and that's to respond non-specifically to a wide range of harmful substances. Okay, so again, the first line of defense is just the skin and the mucous membrane, and that's just to kind of keep everything out. And we'll have more about that on slide 16. And then if something does get in, we'd have to go to our second line of defense, and that involves the non-specific ones, cellular defenses, activities of our neutrophils, macrophages, and natural killer cells. And then I put the slides that that's discussed. Because there's so much discussed in the immune system, it helps if you can kind of keep it all organized and what what specifically is being discussed where. So this kind of helps you. So as you get to those slides 17 through 20, you know, okay, I'm learning now about the cellular responses that are part of the second line of defense, which is part of the innate immune system. So if you want to kind of make a flow chart, or, you know, however you can best organize this information. And then there's chemical defenses, which is part of the second line of defense as well. And those are going to be, they're going to involve cytokines and things like that, discussed on slides 21 through 25. And then the last one is our physiological responses, or fever and inflammation. So all of these different things, and we're going to start talking about these are part of the innate immune system. Once all of these things are already happening, the third line of defense is the adaptive immune system, or the adaptive immune response, and we'll talk about that after the innate response. So for our innate immune response, the first line of defense was preventing the entry of harmful substances into our body. So we have our epithelial layer of our epidermis and connective tissue of our dermis that provides that actual barrier that very few microbes can actually get in, assuming that the skin is intact. Of course, once we cut our skin and we get an open wound, you know, anything is fair game. So this tells you some of the different defenses of the skin. The skin produces antimicrobial agents, such as lysozyme. Lysozyme is kind of like our body's natural antibiotic. And sebum, which is oil. Now, both of those two things are present on the surface of the skin, and they prevent the growth of harmful bacteria on our skin, so that our bacteria don't just settle in and start replicating all over our skin. Harmful bacteria, that is. The skin contains what we call normal flora. Normal flora is non-pathogenic bacteria or non-pathogenic microorganisms, and they do thrive on our skin surfaces, and they help to prevent the growth of the pathogenic or the harmful bacteria. So, of course, we're kind of in weird times right now with coronavirus, but under normal circumstances, that's one reason we don't want to constantly be washing with antibacterial soap or constantly applying hand sanitizer, because you can kill off the normal flora, which is the good bacteria that that helps to prevent the overgrowth of harmful bacteria. That's not to say we shouldn't wash our hands. That's just saying that, again, under more normal circumstances, we don't want to overuse antibacterial hand soaps or hand sanitizers. Regular soap and water is good. We have our mucous membranes. Our mucous membranes line the openings of our body, and they contain something called mucin. Mucin is just a protein, and then it mixes with water to create mucus. So every once in a while you'll hear me use the term mucin and mucus interchangeably. That's just because, I mean, as soon as that mucin hits water, it becomes mucus. So mucus helps to trap microorganisms before they make it further into the body. It also contains lysozymes, or lysozyme, which helps to kill any bacteria that's present there. And then other tracts of our body contain normal flora as well. So your GI tract has normal, healthy gut bacteria that helps to prevent the overgrowth of harmful bacteria. So these are all different ways that we help to prevent the entry of harmful things into our body. So once a pathogen has crossed that first line of defense and has actually gained entry into our body, then we've got to move on to use our second line of defense, which is still a component of the innate immune system. So one portion of that second line of defense is cellular defenses. So these cellular defenses act in a non-specific way, meaning they're not targeting a particular pathogen, but just any harmful thing that would be there. And in this case, it's neutrophils and macrophages, and they work to phagocytize infectious agents. So if you look at the diagram, you can see the infectious agent here, and then the large phagocytic cell is reaching out those pseudopods to phagocytize it. It puts it in what's called a phagosome, which is a little kind of vesicle containing the infectious agent. The phagosome fuses with the lysosome. If you remember from 2401, the lysosome has digestive enzymes to break things down. So those two vesicles fuse to form what's called a phagolysosome. So the digestive enzymes that were once in the lysosome can now have access to that infectious agent and breaks it down, and then you can exocytose the residual particles. So the neutrophils are usually the first ones there at an inflammatory response, so they're kind of the first ones on the scene. But the macrophages, they may show up later, but they typically stay around to make sure everything's cleared out. So neutrophils are typically there first, macrophages typically stay around longer. Another type of cell that works non-specifically against pathogens is our basophils and mast cells. As opposed to the neutrophils and macrophages, these don't work by phagocytosis. These are chemical secreting cells, and the chemicals that they secrete promote inflammation. So typically our basophils are circulating throughout our blood. The mast cells tend to reside in the connective tissue of our skin and our mucous membranes, but both of them release chemicals during an inflammatory response. So histamine is one of those chemicals. Histamine increases vasodilation and capillary permeability. So why would that be important? Well, anytime we vasodilate, we open up those vessels, and increasing capillary permeability allows more fluid, more blood, and ultimately more leukocytes to leave the blood and get into the infected tissue space. Heparin prevents blood coagulation, and then eicosanoids further promote and enhance the inflammatory response. Another important cell type are our natural killer cells, or NK cells. These guys destroy a whole wide variety of unwanted cells, virally infected cells, bacteria cells, tumor cells, cells of transplanted tissue, which is something that we actually don't want to happen, but if we don't do anything to stop it, it will happen. So our natural killer cells are formed in the bone marrow. They circulate throughout the blood. They tend to accumulate in our secondary lymphatic structures in the lymph nodes, the tonsils, and the spleen. And their job is simply to kind of patrol the body in an effort to detect any unhealthy cell, any infected cell, any cell that shouldn't be there, and in a process that we call immune surveillance. Now what happens is once that NK cell has detected a cell that shouldn't be there, it releases these two molecules, perforin and granzymes. Perforin, shown in this diagram by the little blue sticks, comes out and it kind of creates a pore on the unwanted cell. So here's our natural killer cell. Here's the unhealthy or unwanted cell. The natural killer cell releases the perforin and it kind of creates a pore, and that pore opens that cell up. So then the natural killer cell also releases these little spheres. These are granzymes, and the granzymes can enter the unwanted cell through that pore and then get into the unwanted cell. Now, once that happens, the granzymes induce the process of apoptosis to kill the unwanted cell. So the perforin creates a pore, the granzymes initiate the process of apoptosis, so this cell will die. Now it's important that it dies specifically through apoptosis, because in that way it can't spread the infection. If you remember from 2401, there's two ways a cell can die: apoptosis and necrosis. If it dies by necrosis, essentially the cell explodes, and anything that's in the cell can now infect the neighboring cells, so the infection would spread. So it's very important here that it dies via apoptosis, because it then kind of shrivels up, as you can see in the picture, and dies kind of nice and quietly without this big rupturing event, so that we don't spread that infection. And lastly, in terms of cellular defenses, are our eosinophils. Eosinophils target our parasites, so they release these chemicals that are toxic to the parasite. They can also engage in phagocytosis of antigen-antibody complexes. So whenever we have something in our body and then we generate an antibody against it, the antibody will bind to the pathogen to inactivate it so that the pathogen is no longer harmful to us. But then we've got this big complex, the antigen, the antibody, floating around. So we got to clear that out. So eosinophils can phagocytize that. More commonly, however, they get rid of our parasites. So if you're looking at a parasitic worm, so these were one of those non-microscopic organisms, and the eosinophils release these chemicals that are toxic to it, so to try to help kill off any parasites in our body. So moving on to the next component of that second line of defense. Remember, we said the second line of defense has cellular defenses, which we just talked about, and now we're going on to the chemical defenses. So interferons are part of that. Interferons are a class of cytokines, so there's that word coming back, that get released from a variety of cells. And it, again, is a non-specific defense mechanism. So it's not against a particular virus, but just any virus. And its job is to prevent the spread of that virus. So what happens is the interferons get released from a cell that's already been infected by that virus. So in this diagram, here's your infected cell. It's been infected by a virus, and it starts to release interferon. Well, that interferon then travels to the neighboring healthy cells, and it triggers this cell to make enzymes that destroy viral RNA or DNA. So if you remember, viruses propagate by taking over the machinery of the host cell. So a virus wants to get into this normal cell and use its mechanisms to replicate its own viral DNA and RNA. But in an infected cell, it releases interferon. That interferon communicates with our healthy cell to stimulate the synthesis of enzymes that specifically destroy RNA and DNA, so that if a virus tries to infect this cell, it will not be successful. Another thing that interferon does is it stimulates our natural killer cells in our macrophages so that they can kind of be aware that this virus is there, and then they will go and destroy those virally infected cells. So natural killer cells, we talked about how those release perforin and granzymes and induce apoptosis. And then macrophages, of course, are phagocytic cells that phagocytize unwanted cells. So interferon is the cytokine here. It's being released by an infected cell, a virally infected cell, and it works on healthy cells to prevent the synthesis of viral RNA and DNA. It works on natural killer cells so that they will be stimulated to release perforin and granzymes when they find that another infected cell. And it works on macrophages so that they would phagocytize any infected cells. Another important component of the chemical aspect of our second line of defense is what's referred to as the complement system. The complement system is basically a series of plasma proteins that are involved in the activation of immune response whenever we encounter a bacteria or pathogen. The proteins in the complement system are continuously produced by the liver, and they get released into the blood as inactive proteins. So they're made, but they're not active and doing a job until the complement system becomes activated. They get activated by enzymes once something has gained access into the body. There's two pathways involved in the complement system, the classical pathway and the alternative pathway. In the classical pathway, the complement protein binds to an antibody that has previously bound to a pathogen. So let's say we have a bacteria. Our body generates an antibody against it. The antibody will bind to the bacteria, and then the complement protein binds to that antibody. So it's kind of a three-tiered effect there. Now, in the alternative pathway, the complement protein binds to the polysaccharide on the cell wall surface of the bacteria or fungus. So on the alternative pathway, the complement protein is binding directly to the harmful agent as opposed to binding to the antibody. So the complement system activates several defense mechanisms. We're going to go through each of these: opsonization, inflammation, cytolysis, and elimination of immune complexes are all processes that can happen as a result of complement activation. Opsonization is when a complement protein binds to the portion of the pathogen and it increases the likelihood of phagocytosis. So it kind of helps to make it more visible to the immune cells. So here is our pathogen. Here's our complement protein. The complement protein binds to the pathogen, and it's almost like Halen ataxia. It kind of waves down a macrophage. So now the macrophage will come along and phagocytize the whole thing. So opsonization just basically increases the likelihood that an immune cell is going to see the pathogen. Cytolysis happens when we get these multiple complement proteins that form a pore. So it's another pore on a cell. It's different than the pore that we saw with perforin and granzymes. But you'll see these little complement proteins, and they come in and they create this pore on our bacteria. They'll do it over and over. And then when that pore is there, it allows fluid imbalance or fluid disruption. And what you'll get is you'll get an influx of fluid coming into the bacterial cell, and ultimately it will rupture. So cyto means cell, lysis means rupture. The cell is going to rupture. Okay, so here's our complement proteins, here's our pathogen. The complement proteins orient themselves in a way to create a pore that disrupts the fluid balance in the bacterial cell, and then you get this influx of fluid until it essentially lyses or bursts open, and then we've essentially killed our bacterial cell. Elimination of immune complex. Complement protein will attach to an immune complex. So when I say immune complex, I'm talking about an antigen bound to an antibody. Here's our antigen. Here's our antibody. This is an immune complex right there, those two things. So the complement protein will bind to that. Here's our complement protein. It binds to the whole immune complex, and then it can also bind to an erythrocyte or a red blood cell. And then the red blood cell acts as kind of like a shuttle, and it will bring this whole thing to the spleen. Then you've got macrophages that live in the spleen. It could also go to the liver, and it will strip the immune complex and then put the erythrocyte back into circulation. So here's our bad guy, our antigen. We formed an antibody. The antibody binds to the antigen. This is an immune complex. Complement protein binds to that and then attaches it to a red blood cell. The red blood cell will bring this whole thing to the spleen or the liver, and then macrophages there will kind of get rid of the immune complex, phagocytize that, but allow the red blood cell to go back into circulation where it can do it again. So far we've talked about our first line of defense in the innate immune system, which was keeping things out. And then our second line of defense involved cellular defenses, we've talked about those, chemical defenses, we talked about those, and then the last one was physiological responses, so inflammation and fever, which is what we're getting to now. So inflammation is an immediate, it's a local, non-specific event that happens in vascularized tissue, so tissue that has blood vessels, against a variety of different stimuli. So the first thing that happens is you get the release of certain chemicals, and those chemicals cause vasodilation, the opening of those blood vessels, and increased capillary permeability. You get recruitment of immune cells, and you start to get some of that complement activation. The next thing that happens is you get even more vasodilation and the exposure of something called cell adhesion molecules, or CAMs, on the surface of those endothelial cells, which provides a site of attachment for the leukocytes. So the way I typically think of this, and I think I've got a slide coming up that shows it, is if you are kind of on the riverbank and somebody's floating down the river and needs your help getting out, and you reach your arm out to catch them as they pass by and help to pull them out, those are those cell adhesion molecules. They kind of expose themselves from the endothelial cells, which again are the cells of the blood vessel wall, so that as these leukocytes kind of flow by in the blood vessel, they have something to attach to, and that's going to help them move out of the blood and into the tissue space. Okay, and so that was step 3, actually, the movement of those leukocytes out of the blood and into the affected tissue. Okay, so the main effect of inflammation is you get this net movement of fluid that's moving from the blood and into the interstitial space within that affected tissue. That increased fluid then moves, it's going to go into the lymphatic capillaries, and any of that infectious agent that was in that tissue can get pushed into the lymphatic capillaries with the fluid, and now it's officially in the lymphatic system, so it can be filtered throughout the lymph nodes. Okay, so here you can see this poor little kid. He was skateboarding and he scraped his knee. So once he scrapes his knee, that first line of defense is gone. We've broken the skin barrier. So here's the skin right here, and when we break that barrier, all these bacteria can then come and get into this tissue space. Okay, so then we have the release of inflammatory and chemotactic factors. So inflammatory factors we know is going to promote inflammation. Inflammation is going to promote capillary permeability. Look at the lining of the vessel right here. It's solid and intact. Look over here, you have these gaps. Here's a gap in the endothelial wall. Here's a gap. That's increased capillary permeability. Chemotactic factors are factors that are going to promote different cells and other chemicals to come to the infectious site. It's basically like saying, hey, we've got bacteria here. We need all you guys to come to this area. So that happens. So you get the movement of these cells. You've got your mast cells and your basophils, and they're all releasing these chemicals, those little green spheres. Okay, vascular changes. You've got vasodilation of the arterioles, increased capillary permeability, and the display of those cell adhesion molecules, or CAMs. So this little yellow thing right here exposed on the endothelial cell, that's your CAM. So notice that this neutrophil here will attach to it. You see that here. And then now it's kind of lodged there, and it goes through this process called diapedesis, where it basically slips out of the blood and then into the tissue space. So that's kind of the recruitment of cells. So now your immune cells are over in the infected tissue, so they can start to phagocytize or work by whichever mechanism they're going to work best to help clear not only the bacteria, but any dead or dying tissue that may have resulted from it. Okay, so here's some kind of broken down bacterial particles, and then you've got all this extra fluid in here. And remember, anywhere we have blood capillaries, we're going to have lymphatic capillaries. So that all this interstitial fluid that contains the broken down bacterial particles can go into the lymphatic capillary. So now it's in the lymphatic capillary. And if you can kind of think beyond this picture, where's it going to go next? It's going to continue to travel through the lymphatic system, and eventually it's going to hit a lymph node. And there's going to be immune cells in there that see this broken down bacteria and say, oh, there's a bacteria somewhere in this body, and they're going to start another portion of the immune response that's going to create cells that specifically target that particular bacteria. So this kind of just puts it all in a big picture so you can see all the variety of different things kind of happening at once. The signs of inflammation are redness, because you've got increased blood flow to the area, heat, because you've got increased blood flow and increased metabolic activity in the area, and then swelling, because you've got fluids that are moving out of the blood vessel into the interstitial space where fluid normally doesn't accumulate. It can also cause pain, because you've got pain receptors in there, and as that fluid moves out, it starts to compress them and stimulate those pain receptors. And then, so typically what they tell you is to put ice over the inflamed area. The ice will induce vasoconstriction, so it'll kind of reduce the amount of blood flow to the area, so that will in turn reduce the heat and the redness, and can also kind of numb the pain associated with it. Fever is the second and last of our physiological defenses, and it's defined as the elevation of the body temperature by at least 1 degree Celsius from our typically accepted body temperature of 98.6 degrees Fahrenheit. It's initiated by the release of molecules that we call pyrogens. Pyrogens get released into the blood, and they cause the release of something called prostaglandin E2, or PGE2. PGE2 is another example of a cytokine, and when it gets released, it causes the hypothalamus to increase the temperature set point. So our temperature goes up. There's 3 stages of fever: onset, stadium, and defervescence, as long as the infection continues uncontrolled. During fever onset, the hypothalamus stimulates the blood vessels in the dermis of the skin to vasoconstrict, and that decreases the heat loss through the skin. Because those blood vessels are so close to the surface of the skin, if they are open and vasodilated, we can lose heat through the blood that way. But by vasoconstricting them, it conserves our heat, so we reduce the amount of heat that gets lost through the skin. So this causes a person to shiver, to increase heat production through muscle contraction, so consequently the body temperature rises. Stadium is where the temperature just kind of remains elevated, so it's the continued elevation of body temperature. During that time, your metabolic rate increases to promote physiological processes involved in elimination of that pathogen or harmful agent. And then the final stage, called defervescent, this is when the temperature starts to go down. The hypothalamus stimulates the mechanisms to release heat from the body, so including vasodilation. So now we're going to open the blood vessels in the skin. We're going to lose heat that way, and then we start to sweat as well. So we always recommend you increase your fluid intake during a fever to prevent any dehydration that would occur through increased body fluid loss. Fever comes with both risks and benefits. Some of the benefits is that increased body temperature kind of disallows or slows the growth and reproduction of any bacteria in there. But at the same time, it increases our immune system's ability to deal with it. So it increases the activity of our adaptive immune response and accelerates tissue repair. So most physicians would recommend letting a fever run its course and only taking medication to reduce it if it gets too high or starts to cause significant discomfort. But if it's low and is tolerable, then it's actually doing its job and reducing the spread of the infection, helping your body handle the infection. Fever is significant once it reaches 100. Higher fevers, 102, 103, are potentially dangerous because they can cause changes in metabolic pathways and protein denaturation. You can actually get seizures when you have high fevers that are sustained for a period of time. Irreversible brain damage can happen at temps higher than 106 when those are sustained, and then likely get death when it reaches 109. So here's an image kind of showing all the different things involved in this first line of defense. There's some things in here that we didn't actually talk about, but you've got the skin. So the skin obviously is like the barrier, helping to keep things out. You've got normal flora growing on there to kind of curb the growth of pathogenic organisms. You've got sebaceous glands, or your oil glands are going to release oil onto the surface of the skin that also kind of prevents the accumulation of harmful bacteria. Mucosal membrane lining our different tracts, which have normal flora, but they also have mucus to trap things so that it doesn't make its way further in the body. It's got lysozyme, that natural antibiotic we have. It's got antibodies in there, so there's different things within the mucosal membranes to defend our body against something trying to get in. And then you've got, lysozyme and antibodies again in our lacrimal glands, in our tears, and our saliva. So just different ways to help keep things from getting further into our body. This is a good chart to show all of the second line of defenses. So second line of defense had the cellular defenses. We talked about all of these. Basophils and mast cells releasing chemicals. Talks about the different chemicals that are released. It promotes inflammation. Neutrophils and macrophages acting as phagocytic cells to phagocytize bacteria. Natural killer cells using their perforin and granzymes to induce apoptosis in an infected cell. And then eosinophils acting against our parasites. So I would spend some time and go back through all of these, just kind of read the details. It's kind of a summary or a review of all the things that we've already talked about. And then over here you've got those chemical defenses. You've got interferon, prevents the normal cell from replicating viral RNA and DNA. Interferon activating the natural killer cells so it can use perforin and granzymes, activating the macrophage so it can phagocytize things. Everything down here is complement. So you've got opsonization, membrane attack complex, elimination of immune complexes. So go through all of that, all the different options that our complement system can do. And then up here are our physiological defenses. Inflammation, we talked about that, and then fever. So then after all of this is being done, actually at the same time all of this is being done, we're also working on our adaptive immune response. It just hasn't kicked in because it takes some time. So these are all the first things that you're going to see happening in the body.